Okay, this week we are covering cell division, and we're going to do it two ways. It's mainly about mitosis and meiosis. I will show you just briefly what it looks like when bacteria cells divide, which is very simple, and that's actually in our unit. But we did talk about this last week. Talk about this being, we talked about all the cell parts, and I had mentioned this. This is the nucleus. This is the nucleolus of a cell. Right now, you cannot see chromosomes inside of this cell. Chromosomes look like an X, and they're joined together by a central structure called a centromere, and I'll show you that in a second. But right now, you can't see these. And that's part of what this introductory video, and I'll show you some other things as well, but chromosomes are actually in the form of what are called chromatin fibers when they're inside of a nucleus, when they're here. You can't see them. If you were to take some cells out of your cheeks and put them on a slide and stain them, you wouldn't be able to see. You could see a nucleus, you could see the nucleolus. You really couldn't see any of these parts, these little organelles, but you couldn't see the DNA inside of the nucleus. And the only time you can really see that is prior to what is called cell division. So let me walk you through a few steps. Now, we're going to talk about mitosis first. But you see this? It says cell undergoes reproduction or cell division. The two daughter cells are genetically identical, meaning they have the same number of chromosomes as the parent cell. As a result of mitosis, you end up with two daughter cells. One cell, you end up with two daughter cells. So, to get you started, and I apologize for the sound, here's a brief introduction to human cells.
Cells are the basic living units of tissue. The human body is composed of trillions of cells. There are over 200 different cell types. Each specializes to perform essential functions. Bone cells form and regenerate the bones in the body. Nervous system cells send messages within the brain and throughout the body to produce actions. Cells in the blood carry oxygen and help the immune system. Male and female sex cells unite to produce offspring. The genetic information inside each type of cell acts as an instruction manual telling a cell how to function and replicate.
Okay, I hope that helps. I apologize for the sound. We are going to focus on humans for this section. Now, in our PowerPoint, and I'll get to this in a second, there is a pretty cool chart here. It shows the number of chromosomes per body cell for a koala. Here's a deer that has 6. Human beings, we have 46. A buffalo, if I'm reading this right, has 60. Dogs have 78. This rat has 102. So chromosome number does not determine necessarily higher intellect. But human beings, we are 46 chromosomes. That's really what we're going to focus on with this section. Now, in that video, you saw there are different types of cells. This happens to be skin. This is actually, looks like the skin, the top layer of skin, the epidermis of a hand or foot. And all of this, so starting here, all of that is called the epidermis of skin. And then this down here is called the dermis. Skin has two layers, the epidermis and the dermis. The reason I'm showing you this, these are called squamous cells. It's an epithelial tissue. 90% of all cancers are carcinomas, and that would be a cancer of epithelial cells because they divide so quickly. That video had mentioned, or it showed a nerve cell. It didn't mention this, but liver cells, they divide every week. Nerve cells at some point really do not divide. Skeletal muscle cells, muscles that make up your voluntary movements. When you lift weights, you build muscle up, that's skeletal muscle. Those do not divide prior to being, after being born. They're called fixed post-mitotic. But epithelial cells divide pretty often, and skin cells divide daily. Everybody loses skin every day, and then it gets replaced. I say daily, every couple of days. Very high rate of mitosis. Okay, we'll do the cell cycle first, and then we'll do mitosis. Now, before we get into this, it says a eukaryotic chromosome. I'm going to briefly jump back over to our unit. Let me go down here. And I'm just going to touch on this. So this is the reading portion of our class. It's already there. That is showing, it says prokaryotic or prokaryote cell division, which is division of bacteria. You can see, and I mentioned this, bacteria have one chromosome. You see it there. This is called binary fission, and it's very simple. This is not mitosis. You have one cell, and then boom, it divides into two cells. Bacteria can divide as often as every 20 minutes. And in fact, up here it says binary fission. We're mainly going to be talking about eukaryotic cells. Obviously, they're not going to go through meiosis, which is the production of sex cells, but this is how bacteria divide. Like it says here, prokaryotes do not have a nucleus. Their cell division takes place in only two stages. The DNA is copied, and then the cell splits in a process called binary fission. And it's just one circular chromosome. They're very simple, but that's under our overview part of our course. All right, I'm going to go, just while I'm here, just not to waste any time. So let me go back. So we're into Unit 4. This is where we are. All right, I'm going to go down here. So the eukaryotic chromosome, like I was saying, they're called chromatin fibers prior to division. So as a cell prepares to divide, its chromatin fibers coil up, forming compact chromosomes that can be viewed under a light microscope. So a couple of things about this, and this is on your quiz. How is DNA, how is it found inside of a cell? And this is as a chromatin fiber. So I'm going to move down here. This is a good diagram. So here, DNA is double-stranded, and you can see it's coming around here. It is actually wrapped up in these little proteins called histones. And it says here a nucleosome. It's just showing them as in bunches. But if you unwrap the histones, that's when you get the DNA strand. So this is showing, how a chromatin fiber is actually constructed. You have little histones that the DNA is wrapped around. If you uncoil it, now you can see it's a double helix. All right. This is a very key part of both mitosis and meiosis. Before a cell begins to divide, it has to duplicate the chromosomes. And I had found these cells somewhere. They're pretty simple. I like them. This right here is a cell in its normal cycle. It's called interphase, which is not mitosis. This is a cell that's in the very first stage of mitosis called prophase. And I'm showing you this because that is supposed to be representing the chromatin fibers that are not coiled up. This is showing a cell that's about to divide into two cells. And you can see the little X's, and it's got it right here. This is called a duplicated, chromosomes are duplicated. Each side of this, like it says here, is actually called a chromatid. So what you have here in the middle, this is called a centromere. It mentions it right there. A little circular structure. And it's holding these two sister chromatids together. So the way you can read this, human cells have 46 chromosomes. Well, if you're going to split and form two cells, you have to double the number of chromosomes. And like it says there, before it begins, it duplicates the number of chromosomes. So what this is showing, there are 46 chromosomes on this side, and there are 46 on that side. You don't hear it referred to that as much. And sometimes it's called pairs. You have two pairs of 46 chromosomes, but they're called chromatids. So each chromatid during this process will eventually uncoil like that and become chromosomes again when the cell's done dividing. But just remember, if you're going to make two cells, of course you have to duplicate everything, including the nucleus, including all of the chromosomes. So let me back this up. So, yeah, two sister chromatids are joined together tightly at a narrow region called the centromere, which is what that is. So there it is right there. When a cell divides, the sister chromatids of a duplicated chromosome separate from each other. There's a stage in mitosis and meiosis. Meiosis is different. It's got two stages. But right here, these will split. And like I said, this would represent 46, and this is 46. So if you're going to make two cells, this is going to go to one cell. This is going to go to the other cell. So once separated from its sister, each chromatid is considered a full-fledged chromosome. And it is identical to the original chromosome. Yeah, it's duplicated. Okay, the cell cycle. What this means. Now, I gave you, there's all kinds of pictures that represent this. Here's one here. I posted this for you. Yeah, here we go. I posted this. This is a sort of a diagram that's got a whole bunch of just information on it, some really good pictures. What the cell cycle represents is the entire cell life. And starting here, where it says G1, S, and G2, all the way around to this point, that is called interphase, which is not mitosis. G1 and G2 represent growth phases when the organelles are being duplicated. The S phase represents growth and DNA replication. So, in other words, the S phase is when this is happening. You have 46 chromosomes. They have to be duplicated so that the new cell gets 2 each if the cell is going to undergo mitosis. Now, this is in your outline. This one doesn't really have it, but you can see the G1 and G2 here. Those represent growth phases. And then the S phase, it's when DNA, like I said, is synthesized or duplicated. Interphase, metabolism and growth 90% of the time. When it goes to divide, which is this little blue part of the pie represents, that's mitosis. So interphase is the period from cell formation to cell division. The term interphase reflects the idea, well, it's basically a cell undergoing its normal cycle. What this one is not showing, and let me see, I believe I actually have, well, no, this has it. I may come back to this. I don't think I have another. I have those. So these are all really good pictures, but this one has the one I'm looking at. So you have access to this. It's called just a diagram. I have posted under our page. See this M right here? Cells do three things. So this cell is an interphase all the way to this point, and you can see right here, here are the 4 stages of mitosis. Cells do three things. Cells either stay specialized, like a neuron, to a point, or like I was saying, a skeletal muscle fiber. They're called fixed post-mitotic. You don't develop new skeletal muscle fibers. A cell will stay the same. Cells die. It's actually called apoptosis. We don't talk about that in this section. Cell death, programmed cell death, which is actually good. It's programmed cell death. Or they undergo mitosis. So that M checkpoint, and right here, a lot of diagrams will have it here. So a cell either stays specialized, it dies. You know, red blood cells only last 120 days. It'll die, or it will undergo mitosis and then go back around. You know, the new cells will then be an interphase. So that's the cell cycle. So interphase, 90% of the time in the cell cycle, this whole thing is spent in interphase. That's supposed to represent it. Remember, these are centrioles. I still have our diagram pulled up. So remember these little golden, they look like they have rays of sunshine coming off. Here, every cell has 2 pairs of these. There's two of these, and then actually it will develop another two. There's two pairs in each cell. That's what this is representing. Here are the two pairs. When a cell starts to divide, this pair is going to align over here, and this pair is going to go on this side. Okay, so let's, and a key point here, mitosis is division of non-sex cells. So I'm going to go through all the stages. You have to know the order they go in. It's PMAT, P-M-A-T, prophase, metaphase, anaphase, and telophase. And if you're taking a cell biology course, yeah, you can break that down into early prophase, late prophase, early metaphase, late metaphase. That's not our purpose here. So what it is, it's division of non-sex cells. 99% of the cells in your body divide by mitosis. Only sex cells, eggs and sperm, divide by meiosis, which is not mitosis. The rate of mitosis depends on the type of cell. Skin cells divide every few days, whereas other cells divide rarely. Some cells do not die but undergo apoptosis, which is the programmed cell death. I don't know why it says do not die, but apoptosis means programmed cell death. All right, here are the stages. And in your PowerPoint, I gave you some really good pictures, and I'll start here. This is interphase, and so everything I'm about to talk about is listed right here. So, what I did is I put just some very brief what happens during each stage. So I'll do this twice. I'm going to use these first. So here's interphase. Normal 90% of the cell cycle, is what that chart said, is spent in interphase, normal cell activity. See, here's the chromatin fibers. See, they're loose. There's a nucleolus. Here are the two pairs of centrioles. There's the nuclear envelope or membrane. I know that says plasma membrane. It's talking about the cell membrane. Either one, they have different names. Here are the four stages: prophase, metaphase, anaphase, and telophase. How often it occurs depends on what the cell type is. And they actually do not stop in each stage. It's just each stage represents a part of mitosis. So here's the first one. Two things that happen during prophase. Number one, you can see them there. The chromatin fibers will condense and they start to form chromosomes. And the centromere is that little center structure that holds it together. You can kind of see these are called spindle fibers, and they're developing with the centrioles, and the centrioles are slowly migrating over here. But the main two things that happen during prophase, chromosomes condense to form chromatin fibers condense to form chromosomes, and the nucleolus and the nuclear membrane disappear. Like you can see, you can't see a nucleolus here anymore. You can still see the membrane, but eventually it disappears because, yeah, the DNA has to move across the cell at some point, so they can't have that boundary surrounding them. This is metaphase, the second stage of mitosis. This is called the equatorial plane, which means the middle of the cell. See, the centrioles are on each side. Here are the spindle fibers, and then all the chromosomes align right in the center of the cell. And that sort of yellow thing there represents a centromere that's holding the duplicated chromosomes together. So the key part of metaphase is they all line up right in the center. A little later, I'm going to show you a thing called a karyotype. Is when you can actually take these chromosomes and you pair them up into 23 pairs, and you can look and see if there's damage or anything done to the chromosomes. It's called a karyotype. It's really cool. That they get chromosomes from the metaphase stage. Here's anaphase, and what happens during anaphase is these little centrioles, these little spindle fibers will constrict and they pull apart the chromosomes. So remember, these are actually called, now this is a chromatid. A chromosome are two sister chromatids together. Each one of those represents 46 chromosomes in human beings. If it was in a dog, it's a different number. But you see they are migrating towards opposite ends of the cell. That's the big characteristic of anaphase. Yeah, so each chromatid is now migrating across, and it even says here daughter chromosomes, but each one of these would represent a fully formed chromosome. But yeah, they're called chromatids during mitosis. Telophase. Two main things that happen. The chromosomes, daughter chromosomes, chromatids, they will uncoil and become chromatin fibers again. The nucleolus, and you can see it there, it says forming, and then here's the nuclear membrane forming around what will be the nucleus. And this part that's called a cleavage furrow, in cell division, in histology, cleavage refers to cells that divide and get smaller and smaller. Because right now, this is kind of it's not a big cell, but it's really two cells that are still joined together. And then what'll end up happening is these little proteins will constrict, they will pinch off right here, and then you'll have two cells. And that's actually called cytokinesis. When the cell finally divides and forms two cells, it's called cytokinesis. Cell kinesis means movement.
The human body develops, replaces old cells with new cells, and repairs itself with the help of cells that constantly multiply by dividing. The process begins in the cell nucleus. Identical copies of DNA molecules organize into chromatid pairs within the chromosome structure. These pairs are connected to each other at the chromosome's centromere. In the next steps, spindle fibers attach to the centromeres, and the chromatids line up along the cell's midline. The chromatids are then pulled apart. A nuclear membrane forms around the two exact sets of DNA and eventually cleaves. The process through which a cell divides the genetic information and instructions within its nucleus into two exact sets within independent nuclei is called mitosis.
So this is a real squamous cell. You can see that's prophase. See the dark stained chromosomes in the nucleus? There's a metaphase. There is anaphase, see they're being pulled apart. And then here is telophase. There's the cleavage furrow. There, the little chromosomes are still there. They haven't uncoiled. And these are actually what are called, these are fish eggs. They're the white fish blastula. But that's a prophase, that's a metaphase, an anaphase, and a telophase. Part of your lab is you identifying cells in different stages of mitosis. And they're all laid out. So here's like an interphase, prophase, metaphase, metaphase, those two. Anaphase, telophase. So here they're all laid out. So here's prophase, metaphase, anaphase, and then like I was saying, cytokinesis is the final split into forming two cells. Mitosis is prophase, metaphase, anaphase, telophase. Cytokinesis occurs during telophase. There is a term for this when the nucleus gets pulled apart, and that's called karyokinesis. And the control of mitosis, there's a lot of things that control that. We'll get to that later. So there's prophase, see metaphase, they line up right in the center, just like it's saying. Anaphase, see they're being pulled apart, and then there's telophase. Now, cells do a lot of things. When cells are in contact with other cells, it's called contact inhibition. They do not divide that often. Cells divide to prevent becoming too large, because if a cell becomes too big, it cannot get nutrients across the cell membrane at a fast enough rate. The surface area is too big. Cells next to other cells will not divide. And there's actually genes, they're called, there's a tumor suppressor gene that keeps cells from dividing out of control, and something called an oncode gene, which has been determined as a cancer gene. And an oncode gene would actually make a cell divide out of control. But yeah, it's a stop and go signal. All right, this last part, and here, okay. So actually, okay. So we'll talk about cancer now. So cancer cells are when cells divide out of control. They lose their stop and go signals, and it's out of control cell division. Cancer cells usually divide so quickly that they run out of blood supply and they die. And then what ends up happening is a dead cell is then basically the immune system puts calcium inside of the dead cells, and that's usually what cancer screenings are looking for, are dead calcified cells. So that's a cancer cell. You can see it spreading out like that. This happens to be a metastatic type of cancer cell. But cancer is, cell control that is out of control, like it says. They divide excessively and may invade other tissues in the body. So a benign tumor, you've ever heard this term, is a non-spreading tumor. I guess if you had to pick a cancer, as long as it's not in an area of the body that's going to get compromised, these do not spread. Metastatic tumors, malignant tumors, and the term is metastasis. See this? That means they have the ability to spread to other parts of the body. So, cancer cells may separate from the original tumor, and they secrete signal molecules that can actually they do a lot of things. They can make blood vessels form toward them to grow. They can spread those characteristics to other cells. But metastasis is when it spreads to other parts of the body. Okay, I'll start my next video there.